Hi guys, Dr. Gillard here again. I decided to kind of remake last week's lecture on gastric gland secretion and its regulation. It was a little rough, so I spent some time trying to cone it and make it a little bit better and more understandable. And let's make a YouTube video out of it because I think other students will benefit from this as well. This is a very complicated subject, but let's try to get through it. So glands of the stomach, who are they? Where are they released? And how are they regulated? Most importantly, that's the one that seems to get people in trouble. So again, stomach glands, there are three main types of these juice. And when I say juice, I really mean hydrochloric acid. I mean HIP. Remember the hipster? Hydrochloric acid, intrinsic factor, and pepsinogen, which converts to pepsin, of course, and mucus as well comes out of there. So when I say juices, that's what I mean by that. But there's three main types of stomach glands: cardiac glands, fundic glands. Very strong AKA for that is oxyntic glands. I probably should call it, Guyton uses oxyntic glands. Some other medical texts use fundic glands, so they're the same. Remember I said gastric glands? Some authors use that, more European. But oh, what a terrible AKA, because other authors use gastric glands to include all the glands of the stomach, while some authors use gastric glands as an AKA, also known as, for fundic glands or oxyntic glands. So terrible. Stay away from that, you professors out there, teachers, or anybody's watching this. Don't use that. If you want to talk about all the glands of the stomach, say stomach glands. Okay, we said these regions don't match the anatomical areas of the stomach. They were named by histologists and pathologists based on which type of gland the region of the stomach contained. For example, the cardiac region of the stomach contains cardiac glands that only secrete mucus. The oxyntic glands, or the fundic region of the stomach, that's the region that contains fundic or oxyntic glands, and that includes the fundus of the stomach as well as the body of the stomach. Pyloric gland matches anatomically histology and pathology in terms of those concepts, because pyloric glands are only located in the antral region, aka pyloric antrum of the stomach. Okay, everything I just said. Cardiac glands are in the cardiac region, fundic glands, pyloric glands, everything I just said, so you can read that one. Stomach gland anatomy, they all live within the mucosal layer of the stomach. They do not dip into the submucosal layer. Remember, stomach gland has three parts. It has a isthmus, a neck, and a base is the bottom, sometimes called the fundus. The gastric pit comes up and feeds the gland, or that's the beginning, but that's not, the gastric pit is not technically part of the stomach gland. Okay, the gastric pit is made up of surface mucosal cells, which secrete mucus. Remember we said the stomach lining is weird because there's no goblet cells, or very little goblet cells, if any. Why? Because each one of them secretes mucus all by themselves. But once we get into the isthmus, that's where we got stem cells in this region. We got different types of cells here. So that's a stomach gland. That applies to the pyloric and the oxyntic gland. The cells, so many different types of cells that we need to talk about. That's what gets us in trouble. And these cells manufacture the gastric juice, which I said is the hipster, HIP, hydrochloric acid intrinsic factor, pepsinogen, as well as mucus. Here's the real two stars of the show. So we have an oxyntic gland here. Of course, it contains cells we're going to talk about. Stars are, we know the stars, right? Parietal cells and chief cells are in purple here. And pyloric gland is also the star of the show, but it doesn't have these big parietal cells and chief cells, which you probably learned in junior high these days. It has, the star of the show here really is G cells that secrete gastrin, but we'll see that in a second. The isthmus, I just said that's where the stem cells are located. Their job is to replace all the cells of the gastric pit and the surface mucosal cells as well. So those are magical cells. They divide by mitosis into whatever is needed. Okay, I actually started this a little, I didn't mean to go back this far, but what the heck? We'll work our way up here. It's going to make this a little longer than I thought. Stem cells are continuously creating new cells. Just like skin cells, they kind of push the new surface mucosal cells up. Remember we said surface mucosal cells turn over every 7 days. So there's always fresh troops. Remember we talked about the gastric mucosal barrier. Part of the strength of the stomach lining is because the cells are always fairly young up there. They don't live very long. So skin cells about every 25-30 days or so. But these cells of the gut turn over really fast, of the stomach turn over really fast. Okay, now let's get into these glands. So let's talk specifically about the oxyntic gland or the fundic gland. By far the most common type of gland in the stomach. 80% of all glands are of this type. Contains parietal and chief cells. Those are the big ones. Right, there's mucose cells too are important, but they secrete mucus. And what else do they secrete? I didn't add this in here. My students, we went over this. There is one paracrine hormone that they secrete. Remember the mucosal cells? PGE2, prostaglandin E2. Remember that one? It's autocrine and paracrine. It stimulates itself to produce bicarbonate and mucus. And it also stimulates neighbors, so it's paracrine and autocrine. It also, well, we'll see that in a second. We won't get into that. But I'll tell you, it shuts off ECL cells, right? It's not super powerful at that, but it lives in the anatomical fundus and the body of the stomach, makes up the lion's share of the, quote, stomach. You could say it's found in the histological and pathological fundic region of the stomach, or the anatomical fundus and body of the stomach. So you got to be careful with that, and teachers got to be careful writing questions like that. So here's the regions. So this is the histologist's view of the stomach. So the quote fundic region of the stomach includes the anatomical fundus, which is this top part, contains the Megenblas or gastric air bubble, and the body of the stomach here. And then we have the cardiac region is by itself. It just secretes mucus. It doesn't. There's no other hormones or anything secreted. There's no neuroendocrine glands or cells in there. And then the pyloric region is still the pyloric region. They tend to call this the antral region of the stomach, but AKA is pyloric region. Another AKA is pyloric antrum. But this is the distal part. Okay, could say that fundic glands are found in the oxyntic fundic mucosa. They're simple branched tubular glands. They're found in the mucosal layer, we said, specifically between the gastric pits and muscularis mucosae. They are not found in the submucosal layer. Review our anatomy of the stomach. Remember what's weird about the stomach wall? Good. It's got that extra layer, right? Here's this 3 layers instead of 2 layers. But here's the stomach. Here's the pit. Okay, this is a pit right here. These are surface mucosal cells. But here's the actual gland. It starts right here. So there's the neck, body, fundus down here. So this is it's kind of like a double here. They share the gastric pit. All right. And there's remember the layers of the mucosa. I'm not going to go crazy into this. Mucosa layer, simple columnar cells. That's the actual lining. And then we have lamina propria. This is the interstitium, though, right? There's capillaries in here. This is also the interstitium, where cells have to leave the capillary. Food, oxygen, nitrogen, everything has to leave the capillaries to feed all these cells. So it swims through this. So this is lamina propria, is also the interstitium. And then we have a muscular straight layer here called the muscularis mucosae. Notice the glands, none of them go past this layer. Submucosa is down here. This is important because this is where Meissner's plexus is. There's some little yellow dots here. That's going to be a big star of the show. Our box plexus is right here between the circular and longitudinal layers. And yeah, so that's the muscularis layer, muscularis externa. Okay, I'm not going to go too far into the weeds on that. There's another fundic gland or an oxyntic gland. Kind of talked about it already. There's the isthmus body. Just another picture. You can see the parietal cells. You can see the chief cells, strong aka, perhaps more politically correct, zymogen cells or zymogenic cells. Chief cells has been around a long time, so boards are probably going to say, but you should know that aka. Yep. Let's see. Fundic glands produce tons of gastric. I mean, this is the most of the gastric juice. It is all of the gas, well, except for the mucose cells produce help with the gastric. 2 liters of gastric juice every day. Gastric juice moves out of the fundus, and yeah. Okay, here's the famous hipster slide. Oh, I didn't mean to go back this far. Sorry, guys, but this is going to be a little longer than I thought. Here's the hipster. The main component of fundic glands, you can use and that's what Google says, this is a hipster, according to Google. So I guess that's a hipster. But hip is the key here. We said already, hydrochloric acid, intrinsic factor, both come from parietal cells. Pepsinogen comes from the chief cells. Amazing parietal cell. Boy, I went back farther than I thought. Oxyntic cells, aka, here's a strange aka, Delomorphous cell. So hopefully they won't use that on boards, but nevertheless, there it is. Let's see. So oxyntic cell, that's always confusing because oxyntic gland, that's not a great, I think everybody's going to call it parietal cell. Again, they're found in the fundic region of the stomach, found mainly in the neck. Again, its job is to release hydrochloric acid intrinsic factor. There it is again. What stimulates the release of gastric juice from the parietal cell? Now we have another, this is, according to Google, this is a hag, like an old witch, Halloween costume, hag. So hag stands for the 3 ligands, or 3 molecules that are released, or 3 ligands which stimulate the release of gastric juice from a parietal cell. So who stimulates it? And there's receptors for all these too. Histamine, H, acetylcholine, or ACH, A, and gastrin. So this is another key. Could have put a lot more stars here on this. And all of these have receptors to bind to. Here's the receptors, lots of stars here. All medical, chiropractic, physical therapy. Everybody's going to ask questions. You got to know these things. Muscarinic M3 receptors, sometimes just call them M3 receptors. That's what binds acetylcholine, turns on parietal cells. CCK2 receptors, sometimes they're called CCK type B receptors, bound by gastrin. The hormone also turns on parietal cells. H2 receptors binds histamine, very powerful stimulator of acid from the oxyntic gland. Another turner on or turns on the parietal cell. And then we have somatostatin receptor 2, or SST receptor 2. Now this binds somatostatin. This is a turner offer. This turns off parietal cells, so watch out for that one. It's in red here. So make sure you know the receptors, and the ligands are right here as well. Make sure you know who the receptors are and who binds to them. There's a little cartoon. So there's the SST receptor. There's somatostatin, which is released from D cells. We'll talk about the 2 different types of D cells. It turns off. This is a parietal cell. Proton pump is here. This is where the magic occurs to make acid. Turns it off. Everything else is turner honors. There's histamine binding to its H2 receptors. Here's a muscarinic M3 receptor, which binds acetylcholine from Meissner's plexus. Comes from parasympathetic efferent nerve endings, which make up Meissner's plexus. And then there's gastrin. We'll talk about that. Binds CCK2 receptor. Okay, and that's a parietal cell. Kind of cool picture. Where does hydrochloric acid come from? We already said hydrogen, potassium, ATPase pumps, which is the cartoon is right here. Sometimes they're just called proton pumps. Pumps are not always So this one's docked into this intracellular canaliculi here. So that means that the cell's been stimulated. These kind of float around, though. They're not always docked like this. They float around and can be made really quickly by the cell if needed, but they're not always docked and working. Okay, pumps float around the cytosol and they get inserted into the intracellular canalicular system, which again is this canal system. And this would connect, we're kind of upside down here, but this would connect to the lumen of the oxyntic gland. And yep, then it could bubble up into the real lumen of the stomach. Okay, and we know already HAG, stimulated by what? HAG, histamine, acetylcholine, gastrin. There's another cartoon of this. The little red things are the proton pumps, which release hydrochloric acid, HCl, and intrinsic factor. Here's a couple still kind of floating around. So this cell's been stimulated, right, because these guys are already implanted. Got a little cold starting, so apologize. I'm not going to have time to edit out my coughs. So you guys should know this. Carbonic anhydrase is kind of the star of the show. Here's a parietal cell, little square of it. There's HAG, HAG. There's the receptors that bind the stimulators to make hydrogen and chloride. Chloride just goes through channels, nothing fancy. It requires ATP to kick hydrogen ion out here in the lumen of the canaliculi. This would be actually not really the gastric pit lumen. That's not correct, whoever drew this thing. But I wanted to show you carbonic anhydrase because these are common questions for medical students, maybe chiropractic students as well. Carbonic, to make a hydrogen ion, you take water and CO2, which is ubiquitous, and carbonic anhydrase, which is an enzyme, creates H2CO3, which is carbonic acid. Carbonic acid dissociates in the cytosol into bicarbonate, which is kicked out the basolateral surface. Remember that crazy? That's the stupidest word, isn't it? Basolateral surface. That just means the bottom of the cell. Here's the apex is up here. Why they call this some histologists, please you can write in the comments below, why in the world do you call this basolateral surface? Just call it basal surface, or I don't know, because that basolateral sounds like you're talking about the lateral part, and it has nothing to do. It always means the bottom of the cell. But anyway, carbonic acid is made from carbonic anhydrase. It dissociates into bicarbonate, which is kicked out into the interstitium here. And there's what we want, hydrogen ion. So that's grabbed. You need to burn an ATP. You trade it for potassium, and boom, you got hydrogen ion out there. Chloride follows it like a puppy dog to kind of keep things electrically balanced. And there you go. Okay, pH is the lumen of the stomach is very acidic, right? Normally, especially if you had a high protein meal, it can get all the way down to 0.1, according to Ross. More of a medical histology book, which I really like. We use June Kiera. But extremely acid, right? It's formed by the dissociation of carbonic acid. Everything I just said, carbonic acid is made by carbonic anhydrase. Lots of stars. Guys, you know when I put stars, I get excited with stars. That's got a high chance of being on my exam. The target is proton pumps. I see what I'm saying. Carbonic anhydrase. So where do I'm not going to get crazy into the details, but proton pump inhibitors, like Protonics, or there's a ton of them. This is the target. They bind to carbonic anhydrase and screw this thing up so it can't make carbonic acid. No carbonic acid, you can't make hydrogen, and there goes your, you can't make acid. So that's where proton pumps. Remember we said H2 blockers work on histamine, but the reason they work in only 50% of people is because we got acetylcholine and gastrin still stimulating the proton pump thing here. So that's why they don't work. Proton pump inhibitors block the just release of acid, period. They screw up the proton pump because we can't make hydrogen to feed the proton pump. What's the job of hydrochloric acid? The digestion of proteins, of course, but let's not forget the most, arguably the most important job. You have to convert, it's pepsin, the one we need to digest proteins. Hydrogen ion converts pepsinogen, a proenzyme, into the real enzyme pepsin. And that's the one that causes ulcerative or ulcers in the esophagus and PUD, peptic ulcer disease, and such. So we need hydrogen ion to create pepsin. Uh, Hydrochloric acid kills almost anything. Remember, our mouth is really dirty, full of all sorts of bugs and germs. But when we swallow those bugs and germs, they get destroyed in the acid environment. The stomach, except one bug, H. pylori, is a magical bug, very strong. It actually loves that acid. So if acid gets crazy high, with Zoller-Ellison syndrome, for example, it can kill. HP doesn't like that. It can't handle that much acid, but normal stomach, HP can thrive in that stuff. And we remember acid, we talked about GERD and NERD and erosive esophagitis and Barrett's esophagitis and cancer. So this acid can be pesky if it's overproduced. Here's the bug, right? H. pylori. I took all the slides out. Dr. Doe can teach you more about H. pylori. But remember the weird thing? Here's that cartoon character. I was trying to remember his name. I still can't remember his name. It's that little dirty character, and he's got a cloud of dirt around him. Well, consider him H. pylori. H. pylori kicks out ammonia, part of its metabolism. It's one of its byproducts is pneumonia, and it literally gets a cloud of not dirt, but a cloud of ammonia around it, and therefore this can grab hydrogen ion and neutralize it. So he can live in very, very acidic environments. So that's HP. Tough bug. We'll talk a lot about that when we talk about the gastropathies. Chief cells. Chief cells, zymogen cells, peptic cells, gastric chief cells are the AKAs, found in the Fundic gland, more toward the base this time. They kind of live down. We already looked at those. They're a little smaller than the parietal cells. They release a proenzyme or enzyme, or you could call proenzyme is a zymogen. Excuse me. Zymogen. It's pepsinogen is who they release. Remember, we just said hydrogen ion converts pepsinogen into pepsin, and I'm sure I had it on the slide, but remember pepsin. Once pepsin is created, it can auto-convert its own zymogen into pepsin as well. So hydrogen ion and pepsin convert pepsinogen into pepsin. Pepsinogen I didn't forget. It's right here. Pepsinogen is spontaneously converted by pepsin via contact with HCl. The conversion occurs only if the pH is below 5, so there's got to be enough hydrogen ions around. So it takes a little bit of time to to digest that piece of steak in your stomach. It takes time. pH has got to get to 5, and then there's enough hydrogen ions floating around to start converting pepsinogen into pepsin. At pH 3, when the stomach is really acidic, its conversion is almost immediate. And so there's autocatalysis. Once pepsin is born, it can actually self-catalyze itself. It can autocatalysis itself. So other pepsinogen molecules are converted to pepsin, not by hydrogen ion, but by pepsin. So that's kind of strange. Remember, boards like strange stuff, so that would be a good physiology one. Also, it's mainly secreted by chief cells, but not exclusively. It's also secreted by some other cells here we'll look at in a second. There's even some in the urine and in the bloodstream. We're not even sure how. That's a little controversial still, but there are some studies, like a Carroll 2007 study that found elevated levels indicates some certain diseases and cancers. So they're kind of working on that one still. That's not going to be on board. So I'm not even sure if this will be on boards. Medical boards it might be. I'm not sure about chiropractic boards, but let's throw it in here. There are two classes of pepsinogens. There's type 1 and type 2. Type 1 pepsinogen secreted. This is the one, if you see a board question it says pepsinogen, assume they're talking about type 1. It's secreted from chief cells, mucous neck cells of the Fundic glands. and we know that. Type 2 we don't know much about. This is secreted actually by mucous neck cells. And maybe we didn't know this either. Type 1 pepsinogen, it's mainly secreted from chief cells, but the mucous neck cells that live in the oxyntic gland and the fundic region of the stomach, they can also make a little pepsinogen 1. Pepsinogen 2 is made not by neck cells in the fundic mucosa, it's made by neck cells in the pyloric, or in the pyloric glands, so in the pyloric region of the stomach. So type 2 goes further down, more distally, I guess that's how I remember it, in the pyloric glands in the mucosal layer of the duodenum. Okay, we looked at that before. I don't think I need to go to that. The point of this, I put this in here. So here's the mucous neck cells in the oxyntic gland, which is in the fundic region of the stomach, secrete pepsinogen 1, but in the neck cells of the pyloric region, they secrete pepsinogen 2. And they're similar molecules, they do the same thing. I'm not going to go into the biochemistry. This is where I actually meant to start this lecture. Understanding the fundic gland and its regulations. Skull and crossbones are here. This is pretty dreaded stuff, and I'm just looking at my time here. I got class in 40 minutes, so hopefully we can get through this. Alright, there's the hipster. This is a very hip hipster. Another way to remember it, hip is the key. Fundic glands are oxyntic glands. They produce hydrochloric acid intrinsic factor. Pepsinogen, again, we've already talked that, but we haven't talked about how they're regulated, and that's kind of the star of this. There's actually 4 mechanisms that control fundic gland secretion. There's a cephalic phase. Some authors add a neurological phase in here. Then there's a gastric phase, an intestinal phase, Guyton, which is a chiropractic, probably medical school board book as well. And you guys know when I say board book, it means that the board of, for example, a board of chiropractic examiners, they only allow questions from board books. So board books are the key to studying for boards, at least chiropractic boards. I know that 100% because I'm on that committee. I've been on that committee to write questions, and they will not accept a question unless it comes from a board book. All right. So cephalic mechanism. So, and I threw Guyton in here. So Guyton only has cephalic, gastric, and intestinal. Guyton, for an old physiology book, does a terrible job explaining this, I must say. This is actually from Guyton and where we're going. The cephalic phase, I had to add this. Guyton didn't have it. They said the cephalic phase only starts from the brain and the and some other regions here, but it doesn't start there. There's receptors in the mouth and nose where it starts from. Those are afferents. So well, let's look at it here. Sorry, my voice is going. And I see my water sitting over there on the table, and I can't. I'm too lazy to go get it, because then I have to stop this. And yeah, cephalic phase of acid production. So there's chemoreceptors and mechanoreceptors, there's stretch receptors located in the mouth and nose, specifically the oral cavity, oropharynx, nasal cavity, nasopharynx, and in the tongue and in the stomach, of course. That doesn't have anything to do, it shouldn't really have that there yet. Doesn't have anything to do. Cephalic is about thinking about food, tasting food, smelling food, chewing up the food. It's all the books are the same on this. They're responsible for 30% of total gastric juice release. So that's a number. You probably should know that number. Maybe not for my pathology test, but definitely for physiology. So the receptors, these receptors we just talked about, the mechanoreceptors, are stimulated by eating and thinking about food. Well, that's a different. Thinking is a different thing, but chewing, mechanical receptors, they're stimulated, and those action potentials travel to the brain, specifically down the vagus nerve, more specifically down vagal afferents, which are parasympathetic nerves. So they go mainly to the medulla, into a nucleus called the dorsal vagal complex. Some authors don't say that. Some authors, like Guyton, says the dorsal motor nucleus. But there's other interactions. It's complex, which the mechanism is complex, but the real place they go to is the dorsal vagal complex, which includes, here's the subcomponents, the dorsal motor nucleus, the area prostratum, nucleus ambiguus, nucleus tractus solitarius. Thoughts is a different thing, so you can think in seafood. Those action potentials are generated in regions of the cerebellum, amygdala, hypothalamus, but they also are sent afferently to the DVC, dorsal vagal complex, just like these vagal parasympathetics are from the mouth and nose. So they all rise at all these signals come into the DVC, which is right here is the pons, right? The medulla is down here. The DVC is right in here. So all the signals gather here, and then they're sent down vagal efferents down to our friend Meissner's plexus, aka submucosal plexus. So they stem one, and this of course is in. We just saw that one at the beginning. That's in the submucosal layer of the stomach. So we're very close to the mucosal, very close to the stomach glands, and so we have chemical messengers that have to turn on the machinery to make gastric juice. And there's two chemical messengers that come from Meissner's plexus: acetylcholine and something called GRP, gastrin releasing peptide. I don't know why it says two peptides. Should have taken that out. That's a neurotransmitter. This is a neuropeptide. But both of these ultimately cause the release of gastric juice. Both of these, they're not hormones. They're paracrine. You can call them. Some call them paracrine hormones, but they're paracrine molecules. They are swimmers. They diffuse from the submucosal layer, and they swim through the interstitium, find the cells of the stomach glands, and do their thing. Okay, here's a picture again, gastric pit, and then here's our fundic gland. It could be an oxyntic gland. Well, actually, the target's more of the, no, it's not. We'll just say just a regular stomach gland here. We've went over this, but this is part of the enteric nervous system. This is Meissner's plexus, and that's where the vagal nerve fiber, the efferent nerve fiber, releases these acetylcholine and GRP are released from these guys right here. I didn't show the wires, but I could have drawn wires in connecting all the way up to the medulla, but I didn't. Everybody get that? And so, like, acetylcholine is released and it swims, got to swim through the muscularis mucosae, and it swims to a parietal cell and stimulates it. Okay, so get how that works. Alright, so we're going to use this. Put this in your noodles, take a picture, get your iPhones, take a picture of this slide. You got to know everything on this slide, and I'm going to add to this as we go. But for right now, here is the vagal efferents coming in from the medulla, and yeah, we're releasing acetylcholine, 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 and these are the things we're going to be covering what these do. But mainly, the big things, and here's gastrin releasing peptide GRP. These are released from nerve endings. They swim. Remember, I just said they swim. They swim, 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 and bind right there. And that's not ever shown, but that's what happens. Okay, so they bind. There's an M3 receptor in a G cell, stimulates it to produce. We haven't talked about that yet. To produce gastrin, we'll talk about that. But we kind of talked about this. So here's more acetylcholine. Binds to an its M3 receptor here, stimulates the parietal cells to crank out HI, hydrochloric acid, and intrinsic factor. See how that works? We'll learn that some acetylcholine, when it binds like to D cells, it's an inhibitory. That negative means inhibitory. So it shuts off D cells, stimulates G cells, stimulates parietal cells, inhibits this fundic D cell, inhibits this antral D cell. Okay, we'll talk about gastrin releasing peptide a little more. It's parasympathetic nerve endings release this. It's a paracrine. We said that already. GRP, we said that already. Released from Meissner's plexus. Here's what we haven't said. Meissner's plexus is everywhere in the stomach, but it's only released in the antral, the pyloric antrum of the stomach, the antral region. It's not released in the fundic region of the stomach. So make sure you get that one, because I could see myself asking a question on something like that, trying to trip you up. Again, it swims or diffuses. I like to say swim, think of it swimming, but it diffuses through the interstitium, binds to antral G cells. G cells are only in the pyloric antrum, of pyloric glands. Okay, acetylcholine does the same thing with regard to that. It also, the binding triggers the release of a hormone called gastrin. ACH triggers the hormone of gastrin, right? Gastrin is released from G cells or antral G cells. Most people just call them G cells. GRP does not bind to anything else other than these G cells, right? So here, GRP swims, binds to the G cell. ACH does the same thing, binds to the G cell. They both stimulate the release of gastrin. Right there. Gastrin is a hormone. It's secreted out the basolateral part of the cell, wherever that is on this diagram. It swims over to a capillary, jumps in the capillary, in the microcirculation, and it's off. It's in the blood, in the microcirculation in the stomach. And then we'll see here in a second, gastrin has two targets. Might as well tell you now. It can flat out bind to its CCK2 receptor on parietal cell, stimulates acid, intrinsic factor. And if that's not enough, it's got a double mechanism. It swims over and binds to these ECL cells, and ECL cells release histamine. Histamine is the most powerful trigger of hydrochloric acid intrinsic factor. So gastrin flat out directly stimulates parietal cells, and it indirectly stimulates parietal cells by binding to ECL cells. ECLs release histamine. Histamine binds to its H2 receptors. It stimulates hydrochloric acid and intrinsic factor. I think I know why I'm coughing. The wildfires here in California, our building actually smells a little smoky today. The parking lot was smoky. So I think that's what's kind of choking me up here. Mystery solved. Thought it was coming down with a cold, but no. Okay, so everything I said. GRP, we went through that already. Here's another cartoon. So here's the enteric nervous system. There's Meissner's plexus. GRP is released, swims over, binds to a G cell. Here's the lumen. Remember this gastrin. It's not released into the lumen. It comes out the basolateral surface. Swims over through the bloodstream, flat out binds to the CCK2 receptor in parietal cell, binds to an ECL cell, stimulates the release of histamine. Histamine binds to its H2 receptor. So we got a double stimulus of hydrogen. We'll go over that again, but I mean that's it right there. I have official slides on that, because you guys know if I don't have it on the slide, I'm not going to test you on it. Acetylcholine is released from Meissner's plexus, same thing, secondary to incoming parasympathetic vagal impulses. Its job is to get H-cell intrinsic factor flowing, the gastric juices flowing. It also causes the release of mucus, according to Guyton. It can bind to mucose cells as well and stimulate that release. In this situation, this neurotransmitter acts as a paracrine, because paracrines are swimmers. In the submucosa, it has to swim through that a little bit, and it, just like GRP does, it has to go through a little submucosa. Why submucosa? Well, that's where Meissner's plexus is, in the submucosa, and through the mucosal layer of the stomach. So via paracrine action, it swims, binds to, and stimulates the following: parietal cells and G cells. Okay. It also inhibits Fundic D cells, or you could say oxyntic D cells is probably better, Antral D cells. So let's talk about this more. Acetylcholine and parietal cells, again, ACH binds to M3, muscarinic M3 receptors. They live, that's its docking site on parietal cells. The binding directly stimulates the release of gastric juices. hydrochloric acid intrinsic factor, i.e. the gastric juices. Okay, we've already showed you that, but we can show you it again. So there is the acetylcholine be released. You got a piece of food in your mouth. We go through that afferent vagus to that nucleus in the medulla, comes back efferent vagus, ends up in Meissner's plexus. The nerve releases acetylcholine and GRP. Same story. They both stimulate the G cell. Hormone gastrin is released, binds to the parietal cell, binds to the ECL. ECL release histamine, double binding, double stimuli of the parietal cell, and boom, hydrochloric acid, intrinsic factor are released. Gastric juices are flowing. What about chief cells? We always forget about chief cells. ACH binds directly to M3 and M1. I'm not going to go down that, but it can bind. There's M1 receptors too. Binds to chief cell M3 receptors. That stimulates the release of pepsinogen. Remember, parietal cells and chief cells both live in oxyntic glands in the fundic region of the stomach. Think about that statement, because sometimes we forget the big picture. Okay, acetylcholine and G cells. I just got through telling you that one. Bind to M3 receptors on G cells. Its binding directly stimulates the release of gastrin. Ultimately, gastrin will cause the release of HCl and intrinsic factor. I just showed you that double mechanism. Therefore, you could say that ACH directly and indirectly stimulates the release of hydrochloric acid. Intrinsic factor, just talked about that, but let's put it in stone here. Gastrin is not a paracrine swimmer. It has to swim a little bit, but it travels through the bloodstream, so it's a hormone. Can travel great distances. It's the most important regulator of gastric acid secretion, period. So that's a good point there, lots of stars. Gastrin is released by G cells. Some people call these antral G cells. To remind you, on boards is not going to call them antral G cells because that tells you where they are. They'll just call them gastrin cells or maybe G cells. Boards don't like to abbreviate. They like to give full words, so maybe they'll call them gastrin cells. Are only found in the antral region of the stomach. Therefore, it enters the bloodstream from the anterior region of the stomach. G cells release gastrin in response to, there's a great question. G cells release gastrin in response to blank and blank. I can see my question now. And it's acetylcholine and gastrin releasing peptide. Gastrin has two targets. We know that already. I went through that a couple times now. Parietal cells, CCK2 receptors, causes release of HCl. The gastric juice, HCl and intrinsic factor, binds to enterochromaffin-like cells, ECL cells, via CCK2 receptors, causes the release of histamine. What does histamine do? We just told you, but let's tell you again. Histamine has the H2 receptors on parietal cells, so it swims over and binds to them. That also stimulates our gastric juices to be released. Who are the gastric juices? Hydrochloric acid and intrinsic factor. And again, histamine is the strongest stimulator of HCl and intrinsic factor. Okay, we've looked at this. I don't think I need to go over this again, but you can look at it, nothing new. What about gastrin in chief cells? Let's not forget the chief cell. There is some research to indicate gastrin also binds to the chief cells, and therefore it stimulates the release of pepsinogen 1. Gastrin is interesting because it causes it's a hormone, it causes growth and development. So it stimulates the growth, the thickening of the fundic mucosa, and can increase the size, the mass, and even the number of parietal cells and ECL cells. Its target, remember, gastrin binds to parietal cells, binds to ECL cells. And so in conditions that oversecrete gastrin, ZE syndrome or Zollinger-Ellison syndrome, people have way too much gastrin. Their parietal cells are always working, ECL cells always working like bodybuilders. So they pump iron and they get massive and huge, and they I don't know, they get married and have babies. So there's too many of them. So they proliferate. So there's too many parietal cells, too many ECLs, and they're all big bodybuilders. So it makes the gastric mucosa really thick, amongst other problems like fistulas, and there's all sorts of problems with this. People with ZE syndrome. Here's a normal stomach. My students know that pretty good. Those are the nice rugae. Everything's beautiful. Here is not so normal. Close up. These are way too big, way too thick. So that's a patient with Zoller-Ellison syndrome who has many, many years of gastrin levels that are way too high. And his parietal cells and ECL cells, which make up this tissue, they're way too many and they're way too burly, too muscular, too thick, hypertrophic. You guys know I like my pictures, don't I? Here's just another antral G cell, just another way to look at it. We're going to talk about another stimulator. We know ACH and GRP stimulate antral G cells. Protein in the stomach, broken down protein. There's probably a couple amino acids, some polypeptide chains. They can also, it's believed, stimulate antral G cells to also release gastrin. And again, we know what gastrin does, so I don't have to go over this, do I? You can just follow the cartoons here. I've went over that enough, I think. Yeah, so protein fragments, as I said, in the antral region of the stomach may also stimulate gastrin. It's believed these protein fragments directly bind to receptors on G cells or antral G cells. No sense calling it antral, that gives it away, as I said. And their apex, remember the apex is the top of the cell that could communicate with the lumen, and that could directly cause the release of gastrin as well. So recap, there are three things that stimulate antral G cells to release gastrin: acetylcholine, gastrin releasing peptide, and now polypeptides or peptide fragments or protein breakdown products as also. So perfect slide question, right? 4 out of 4. Which one of the following is not or does not stimulate antral G cells to release gastrin? Okay, we talked about turning on. What about turning these things off? Let me see how am I doing on time. Oh my goodness. Got to go to embryology in 17 minutes. D cells. So D cells, not the cells, D cells. So they're just called D cells. There's some AKAs for these called delta cells and the symbol delta cells. Don't use delta cells for gastric delta cells, the ones in stomach glands, because there's delta cells in the pancreas. The word delta is reserved for pancreas. So authors and teachers asking questions, this is one time where you have to abbreviate, call them D cells. Okay, there's two flavors. There's oxyntic D cells that live in the oxyntic region. In other words, they live in oxyntic glands or fundic glands. And there's antral D cells, which live in the pyloric region of the stomach, or which live in pyloric glands. These AKAs authors are horrible about flipping these around, so these are just AKAs you have to know. Oxyntic D cells, everything I said, are found in the oxyntic region. Antral D cells are found in the pyloric region. Both of these cell types are considered neuroendocrine cells. First time we talked about there's a bunch. There's some we're not even going to talk about. But these are neuroendocrine cells. So these guys can be turned off. They are turned off by acetylcholine. Remember I showed you that before. But what do they do? If they turn off, they don't do anything. But when they're turned on, what do? They secrete somatostatin, SST, right? That's a flat-out hormone. SST, everything we've talked about so far, acetylcholine and GRIP, GRP, those are stimulators. This guy's a turner-offer, so it's an inhibitor. Somatostatin is a turner-offer of gastric juice. Turner-offer of other hormones in other part of the bodies, including growth hormone, but we'll talk about that maybe in endocrinology class. So despite their similarities, the D cells of the fundic and antral region, there are some major differences between those we need to talk about. Some fun facts in general about somatostatin. It is the most important negative feedback inhibitor for gastrin and gastric acid secretion. So we'll talk about this in a minute, but negative feedback inhibitor. Originally discovered as a hypothalamic peptide, that inhibited growth hormone a long time ago, and then later it was discovered, oh, hey, it's in the stomach. And then it was discovered, hey, there's a different type of it in the fundic region compared to the pyloric region. So also discovered there's delta cells in the pancreas that secrete somatostatin. We're not talking about those now. We're talking about stomach gland D cells. Specifically, let's talk about the antral D cells first. These are involved in that negative feedback inhibition, unlike the fundic region D cells. Live in the antrum, pyloric antrum of the stomach. Their tops, or the apices, are in contact with the lumen. They have little receptors that are tasting the luminal fluid to see how much hydrogen ion is floating around there. Do we have pyloric glands and chief or pyloric cells and chief cells in these pyloric glands? No. How's the acid get in there then? Well, it's just from the general acid of the stomach lumen. Some will drip down into these pyloric glands, and it's tasted by the antral D cells, so we can know how acidic the stomach is. So they have hydrogen ion receptors that can trigger response once the density of the hydrogen ion is sufficient to stimulate them. And what is their response? Once the hydrogen ion gets strong enough, they release somatostatin, or SST's abbreviation, into the interstitium. And then SST is a double. It acts as a hormone, can get in the bloodstream, and a paracrine. So that's kind of weird. What triggers, lots of stars should be on this slide, what triggers the release? What stimulates antral D cells to release SST? Well, it's the amount of hydrogen ion in the stomach. Once hydrogen ion gets too much and the pH goes below 3.5 is the magic number, the D cells are stimulated to release somatostatin. And once released, SST finds its receptor on G cells and binds to it and shuts it off. So this is the first time we've seen a shutter offer of G cells. And we know G cells secrete what? No, not histamine. What do G cells secrete? What's G stand for? Gastrin. They secrete gastrin. So SST is inhibitory. Binding of antral SST to a G cell is inhibitory, turns off gastrin. If you turn off gastrin, what happens to the stomach juice? What happens to acid? What happens to pepsinogen? What happens to intrinsic factor, for that matter? Turned off, right? Turn off gastrin, because remember, gastrin had a double stimulator. It was a powerful stimulator of acid. So we just majorly shut off acid. It also, it's a hormone as well. This is weird. You may not know this, some of you people. SST, antral SST, not oxyntic SST. It can get, antral SST jumps in the bloodstream, and it's transported all the way over to the fundic region of the stomach and dumped out, and it swims over and helps to inhibit ECL cells and parietal cells by binding and shutting them off. Okay, so that's an important difference between antral and oxyntic D cells, and antral versus fundic somatostatin. Got that? Okay, so what are we showing you here? So we're showing you right here. So acid is building up. So this is the lumen. We'll pretend this is the lumen of the stomach, and hydrogen ions are building up. These parietal cells are cranking out acid. Hydrogen ion is building. It's getting more and more, and finally it's enough. There's enough hydrogen ion to make the pH 3.5. That's enough to stimulate the D cell in the antral region. Antral D cell says, okay, that's it. I'm releasing my hormone. It releases SST, somatostatin, and somatostatin gets into the bloodstream. But let's do this first. SST paracrine style swims over to the nearby G cell and turns it off. So no more gastrin, no more binding to parietal cells, no more gastrin binding to ECL cells. There goes a huge part of the stimulus for acid release. But it also goes into the bloodstream, and it swims over and it binds to ECL cells, turns them off. So that's another way to turn off ECL cells, and it directly can bind to parietal cells and turn that off as well. Okay, everybody good with that? So that, what we just described here, hydrogen ion being produced, and when the product of a gland actually induces its own shutdown because hydrogen ion builds up, shuts down, stimulates D cells, SST, etc. That's called a negative feedback inhibition. Very important, high board stuff, right? You got to understand negative. We see examples of that in endocrinology all the time. So antral, not fundic or not oxyntic D cells, but antral D cells and their SST are an important component of this negative feedback inhibition loop, which governs parietal cell secretion. Again, specifically, once the stomach acid is lower than 3.5 or 3.5 or lower, it stimulates the production of SST. And yeah, so that's everything I explained there. I thought I had a picture of that coming. Therefore, SST is the most important negative feedback inhibitor for both gastrin and hydrochloric acid secretion. Good test question, right? What's the most important negative feedback inhibitor? That would be, even more specifically, I should really say that is antral SST. Antral SST. Way more powerful too than PG2. We talked about PG2 shutting down ECL cells. So here's a nice picture of this. So here's parietal cells cranking out their acid. Everybody's great. But once the acid gets too high, it stimulates a D cell right here. So acid is being cranked out in the stomach. Too much of it builds up. Once it gets to 3.5 pH, D cell goes, that's it. We're turning off this stuff. It's going to wreck the stomach. So turn off that acid. Releases SST, binds to gastrin, and then there goes your gastrin. Is a huge stimulator, right, of hydrochloric acid. This is all shut down. So this is a negative feedback loop. Oxyntic D cells, kind of running out of time here. So they're found in the fundic region or oxyntic, or in the fundic region of the stomach, i.e. the anatomical fundus and body. These guys have really long cytoplasmic processes, which are so long they almost are like wires directly connecting to parietal cells, chief cells, and ECL cells. So they don't use a hormone mechanism because they really don't need to because of these long processes. These processes are not long enough to go over to G cells. So oxyntic D cells and its oxyntic somatostatin do not turn off G cells. Okay, remember that. It's pyloric. SST does that. Okay, another thing, oxyntic D cells don't care about pH. They don't have any hydrogen ion receptors like the pyloric D cells do. Remember, they're tasting the stomach lumen. These don't have that. They don't care what's going on out there. So they don't participate in this hydrogen ion negative feedback loop. Okay, so that's an important concept. Well, then what stimulates them? Gastrin. When the levels of gastrin in the blood get too high, these D cells can sense that, and gastrin can bind to them and turn them on. Now this is in Holt, which is one of, I really like it. It's a medical endocrinology book. Not a great storyteller, but James is probably better at that. But anyway, yeah, so it is the elevated blood levels of gastrin will stimulate the oxyntic D cells, turn them on. When they get turned on, they release their oxyntic SST. The shutoff point of serum gastrin occurs, I'm not going to give you the blood serum level, but it occurs about the same time that the pH reaches 3.5. So about the same time as pyloric D cells kick on, oxyntic D cells kick on as well, but it's just a different mechanism. Okay, so make sure you guys get that right. Running out of time. Oxyntic D cells, when stimulated, release SST. We said that. It swims through the interstitium, through those long, it doesn't have to swim far because of those long processes. It binds to ECL cells, parietal cells, shuts them right down. It's always inhibitory, right? It always turns things off. We've already said that. They're stimulated by some other things. So catecholamines can stimulate these. Epinephrine, norepinephrine, dopamine. Epinephrine, the tiger's chasing you. Once those levels get high enough, oxyntic D cells are stimulated to produce somatostatin, and that shuts off the machinery, right? If you're running from a tiger, you don't want acid. You don't want your machinery. Remember, that's an ATP powered event to make acid. We want to save all the ATP for the muscles and brain to get away from the tiger. So D cells are activated by catecholamines. That's oxyntic D cells. Who turns off the D cells in somatostatin flow? Both fundic and pyloric D cells are inhibited, as we said before, by acetylcholine. So if there's no tiger chasing you, you've just ate a meal, you need to release gastric juice to get everything working. And so you don't want the inhibitor, you don't want somatostatin flowing, right? You need that shut off. So acetylcholine not only turns on parietal cells and everything else we talked about, but it binds to D cells and turns them off as well. Pretty simple, right? So here's acetylcholine. We showed you this way back at the beginning. Turns that off. There's gastrin levels can regulate that as well, but acetylcholine is the big one there. And over here in the antrum, acetylcholine and something called VIP. We're not going to talk about that. That's beyond the scope, I think, for our chiropractic boards. Or maybe we'll talk about it if we get to the intestine. Okay, that's basically it. The gastric phase, everything applies. I mean, we cover what we need to cover. The gastric phase accounts for about 50% of acid release. Remember, the cephalic phase was 30%. We're not going to talk about the intestinal phase. I don't have time. That's not really that important. 80% is covered by the cephalic and gastric phase. Gastric phase occurs when food actually hits the stomach and stretches out and contacts the stomach wall. pH starts to rise in response. Hydrogen ions the stomach juices are being released. But once the pH, strike that. Once you get food in your stomach, food is basic, right? Even if you have some acid in your stomach, that food is going to dilute it. And some water, you're drinking water, so the pH goes way up to 7. Once the pH rises above 3.5, well, we said 3.5 was kind of the magic number. So that once the pH is below 3.5, D cells inhibit the release of gastrin. But once it goes above, gastrin secretion is no longer inhibited by somatostatin. And so now gastrin can flow and get the acids going again. So that mechanism by D cells, especially the antral D cells where it tastes the pH, is super important for turning on and off acid. GRP then and ACH will be released because of the stimuli of the cephalic and the gastric phase. The stretch of the stomach stimulates the release of these, and you know the rest of the story. Stomach distension, especially if there's proteins and acid, is a very strong stimuli for the gastric phase. We're not going to get into all the long and short reflexes, but the bottom line is acetylcholine and GRP are released once you get some stretch in the stomach. The stretch activates mechanoreceptors. And I guess we are going to talk, there's the long extramural reflexes and short reflexes. The bottom line with these reflexes is they quickly stimulate Meissner's plexus to release grip and ACH, and you know the rest of the story. ECL cells, we talk quite a bit about them, but they're enterochromaffin-like cells. Be careful because there's an enterochromaffin cell, which we're not going to talk about. These are EC, they're like cells. One of the main subclasses of neuroendocrine cells. Sometimes they're called enteroendocrine. They make up 70% of the total neuroendocrine cells in the stomach. The other two neuroendocrine cells we've talked about, G cells and D cells. What are the three neuroendocrine cells? ECL cells, G cells, D cells. So very important for acid production, as we've talked about, right? What do they release before we go to the slide? The strongest stimulator of hydrochloric acid? Histamine. They live deep in the base of the oxyntic and pyloric gland. They don't connect with the lumen, so they're not tasting the luminal fluid for anything. Their job is to synthesize and make histamine. Uh, the histamine, of course, stimulates. Oh no, they're what stimulates the ECLs. Gastrin and ACH both turn them on. Okay, that's important to know. Who stimulates ECL cells to make histamine, which is the most important releaser of stomach acid? Gastrin and ECH. ECL cells therefore have CCK2 receptors for gastrin and muscarinic M3 receptors for acetylcholine. I can make questions out of this slide, can't I? Who turns them off? They're inhibited by PGE2, which is released by surface mucosal cells, and somatostatin, which is secreted from where? D cells. Okay, there's one right there in the oxyntic gland. Are they in pyloric glands? They're not in pyloric glands. There's no reason for them to be there. We don't need histamine secreted way over in the antral region, right? What good would that do? ECL receptors, CCK. This is kind of interesting. Mr. Spock is curious. I got like I want to be late for class, but that's okay. Be a little late. Interestingly, their CCK2 receptors can also bind CCK, cholecystokinin, which is released by enterocytes of the duodenum. So CCK can, from the intestine, here's the intestinal phase coming in, which we don't have time to talk about. It has the ability to bind to our CCK2 receptors on parietal cells, and therefore they can release a little histamine and can stimulate the release of a little gastric acid. It's not super strong mechanism, but they can. There's also CCK1 receptors that can bind CCK exclusively. That's who they're supposed to bind to. CCK binds to CCK1 receptors. Therefore, gastrin can bind a little bit to the CCK1 receptors. And those are located, remember the targets, gallbladder and pancreas, right? They secrete bile from the gallbladder and they release digestive juices from the pancreas. CCK does that. But technically CCK2, those receptors can bind gastrin and do that a little, but that's not an important mechanism. Okay, we talked about excessive gastric exposure before. ECL cells can be hypertrophic and proliferative, right, with conditions like ZE syndrome. Excessive blood concentrations of gastrin can overstimulate parietal cells. We talked about that. And ZE syndrome, for example, those patients suffer peptic ulcer disease and GERD. Okay, we did it. I got, how many minutes? I got like 4 minutes to get to class, so I hope you enjoyed that. Yeah, good luck on the test next week.